Okay, can somebody please confirm you can hear me? All
We can hear you.
right. I see my picture is crooked. When I show you my screen, is it crooked? Oh, good. The lecture page is finished, so my computer will speed up. Nobody commented, so I'm going to assume the lecture is normal. All right. Um,
We just only see you, Professor.
I know, but I share my screen. Is this crooked or is that straight?
No, it's correct. It's straight.
Okay, good. Like I said, I don't know what's going on. I've got to go into Clark to have them look at this because all right, let's go here. So what I'm going to be talking about today is the unknown project, and you can get those files that I'll be looking at. Are you seeing my screen now? All right, you
We see
can,
you going through the canvas.
yeah, you can get the files I'm talking about here. The one I'm going to start talking about is the unknown instructions. They're there, although we will talk about that one and probably not that one, but this one, and that one. We'll talk about all of those today. So today is February 4th, and what we're going to talk about today is the unknown project. We'll actually start the unknown project today. Before I begin, are there any questions about anything?
I guess I'm just want a bit of a clarification. As we begin this unknowns project, are we walking through the tests we would normally run as if we were in the lab to discern and figure out our organism? Is that how this is working?
Yes.
Okay,
All
thank you.
right, let's go on. So, the unknown project is a 75-point lab project where you will be given an unknown number, which will represent an unknown bacteria. The Unknown Bacteria Project is a time-honored part of most microbiology courses. You're not going to be doing this project in fact because everything we do will be virtually. It is hoped that we'll challenge your knowledge and skills in performing laboratory techniques. Like I said, I will give you a number which will represent a species, and only I will know what species you have. And the different species you can have is actually shown here, which is the unknown list. Let me see if I can show that. So that's right here, unknown list, spring 2020. Same list we've had since spring 2020. And you have gram-negative cells here, I don't know, 11 or 12 of them, and gram-positives here. And these are the species, which one of them will be represented by your number. And you're going to be performing tests for the rest of the term or most of the term, trying to identify your unknown species. For example, uh, I am going to choose the first three tests that you will run. And it'll be the gram stain, the gelatin hydrolysis test, and the PR lactose test. The reason why I did the gram stain, almost everybody starts with the gram stain because it easily divides all of your species into two, either gram negative or gram negative. I want to do the gelatin test early because this test takes the longest to incubate. You have to run that test for 1 week before you'll get results. And so you're going to start that test today. I'm not really going to send you the number tonight, but we're going to pretend that I'm sending you the number tonight, and you're going to pretend that you're starting the gelatin test. You should also pretend that when I give you the species, that you're going to pretend to streak it out for colony isolation. Take cells from an individual colony, streak it out again, and then take cells from two colony isolation events so that you will have pure cells for your stock culture, and you'll use your stock culture to run all of your future tests. Now, in reality, like I said, it's only going to be pretend. That's what you would do if you were in the real lab. We don't have anything that you're really going to do other than that I'm going to send you the number. And I'm just going to send you the number next Tuesday when I send you the test results for the gelatin test, which you're going to start today. And so you wouldn't get your results until next Tuesday. And you're going to start the gram stain on Sunday, where you're going to put the slide in your drawer and you're not going to look at it until Tuesday. And then I'll send you the results of the gram stain on Tuesday. And you're going to start the PR lactose test on Monday. And I'm going to send you the results on Tuesday. The reason for doing all this is that means I'm going to send you one email instead of 4 emails. And that reduces my contact with you. This project, there's 40 students, and assuming that there's a 5 or 6 tests you're going to run, that means there would be something like over 200 emails between me and the students for one project alone. And I just said, that's too much for one project. And I asked the other microbiology instructors when we went all online during the pandemic, would it be all right if I chose the first 3 tests and then I would reduce the number of emails by, let's see, 40 students times 3. Actually, that's something like 80 emails, maybe even 120, something like that. So let's say 60 to 80 emails, which will then mean the rest of the contact, not really an email, but it could be, would be that many times, which is doable. Not ideal for one project, but it's doable. And so that's why I'm sending you the first 3 tests. I just chose them. The gram stain I chose because it's a good one to start with. The gelatin hydrolysis test is a good one because this one takes a week before you will ever get any results. This test takes the longest to incubate. And the PR lactose test is just a good one, so I chose it. That one, no other reason. Um, After you get the results from this and you make your conclusions and you figure out which possible species you have, let me go through. Let's say they're gram negative, so you narrowed it down from all the species now to only half of them. These you remove. And let's say it's gram gelatin hydrolysis negative. That narrows it down from all the gram negatives to, let's just go like here, to this one minus that one. And then it's AG in the PR lactose test. Narrows it down to 4. Now, admittedly, yours might not be narrowed that greatly, but it will be narrowed down somewhat. And then you have to pick a test to figure out which of these 4 it would be. And all future tests, you will pick yourself. You will do that in your unknown notes page. I'll talk about your unknown notes page in a little bit. You could pick the 6.5% sodium chloride test, and actually, if it's positive, that would narrow it down to only 1, and so you've identified your unknown. However, it's negative, you haven't narrowed it down at all because a V means that it's variable, and that means it'll fit both a plus and a minus. So that if it's V, it could fit the plus. And like I said, if it's plus, you got to narrow down to one. But if it's negative, you haven't narrowed it down at all. You have the same four to look for. And you could do this one. It's a little better than that because you narrow it down to 2 and 2, but only if it's a plus. If it's a negative, you don't narrow it down at all. The same with this one, or similar with that one. This could be a possibility. If it's a plus, you narrow it down to one species, but if it's a negative, you narrowed it down to three of the four. A better one would be the methyl red test. Why am I not seeing that? Which, if it's plus, it narrows it down to this one and that one. And if it's negative, it narrows it down to these two. So the first three tests I'll choose for you. The next test you have to choose. And this project is supposed to be doing dichotomous logic. And so you will, after the first three, do each one test at a time. In actuality, with the first three, you are doing three tests at a time. You're starting the gelatin hydrolysis test tonight. You won't start the gram stain until Sunday, and then you won't start the PR lactose test until Monday. It's just that I'm going to send you all of the results at one time. So instead of sending you 3 emails, I'm going to send you one. And then I've got to send you the unknown number, and I'll just send you that at the same time. So instead of sending you 4 emails, I'll send you one. And that just reduces the contact between me and my students. And like I said, this project has a lot of contact between you and me. You will probably have to run about 5 or 6 tests to identify your unknown, depending on how lucky you are and what species you get. Any question about any of that? All right, so I'll send you a number. It'll represent a species. I will then send you, I picked the 3 tests. You will run them at 3 different dates, but you're going to get the results all in one day. And then after that, you will have to put in your unknown notes the results I sent to you for these 3 tests, the conclusions you make for these 3 tests, and then the species under consideration for these 3 tests. Then you will look on this table and pick another test. Assuming you're using dichotomous logic, you will pick one test at a time, request that test in your unknown notes page, and wait the required amount of time for the test to run, meaning there's going to be an incubation test for each test. And then I'll send you the results. You need to read about the tests, and you can do that by scrolling down your web page. Let's go down here a little further to week 5 lab, go past the week 5 lab. And that's for Thursday. Go down to week 5 lab unknown test. And there's all the tests telling you the information. Let's go to the gram stain because I need to talk a little bit about that. The gram stain. Normally you get the results for this test right after you do it, but this time you're going to be doing it. Let me put that over there. You'll get the results on Tuesday, although you're running the test on Sunday. You're just putting your slide in the desk and you're going to look at it on Tuesday. It will give you whether it's gram positive or gram negative. You can also get the shape, whether the cell is a sphere, a cocci, or a bacilli, or a spirilla. I might also send a coccobacillus, but those would be the shapes I could send. It's possible to get the cellular growth arrangement, but we've got a warning here about that. In practice, it's difficult to determine from the gram stain whether the cells are clusters or chains, because the chains can be broken apart into individual cocci, and then the cells can clump together on the gram stain so that what's actually a chain can look like a cluster, meaning look like a staphylococci instead of a streptococci. And the cells can be broken up because the gram stain is a fairly harsh procedure, and the take-home messages do not rely on the cellular arrangement you get in the gram stain. You need, if you want to know the cellular arrangement, to run the cellular growth arrangement test, which you can get right here, cellular growth arrangement test. If you want to run that test, what you're going to do is simply grow your cells in a liquid culture and then take the cells out of the liquid culture and make a wet mount. You can add dye, and then look at the cells in the wet mount to see the cellular arrangement. Are they single cells, diplo cells, staphylo cells, staphylococci? Are they streptococci or some other arrangement which isn't mentioned there? But um, that's how you do the cellular growth arrangement. You grow your cells in a liquid culture and then make a wet mount, which is very gentle and doesn't break up the streptococci. And because it's a wet mount, you don't have the clustering or the clumping of cells, where a streptococci, if it clusters in the gram stain, will look like a staphylococci. Any question about any of that? All right. And you need to look on these to determine the time that the cells need to incubate. Like the catalase test is one of the few where you get the results as soon as you run the test. So if you ask a catalase test in your unknown notes, you might get the results that day. However, if I've already sent out the results for that day, you won't get the results at least for another day. I will promise to look in the unknown notes pages about every other day. Oftentimes I'll look every day, like on Tuesdays and Thursdays, and probably Wednesday, I'll look every day. But over the weekend, I will not look every day. Particularly on Sunday, I will not look at the unknown notes page to see if anyone's put in a test request. So you might be out of luck if you request a test on Friday and I've already sent out the results on Friday. I may not see your request until Monday. And so just realize that when you're putting in a test on Friday or maybe on Saturday, I might not see it until Monday. Okay? Any question about any of that? All right, let's go back to another test. Like I said, the catalase test is unusual. There's only a few tests where you can get the results on the same day you run the test. Most of the tests take 2 days to run, like the citrate test. How long you incubate it is usually in the procedure. This is talking about the citrate test, talking about what the results mean, blue color with growth, interpretation, score is positive. How you do the procedure, and then place the tube in the rack for 2 days at 35 degrees. Okay, so you won't get any results for at least 2 days. And it could be 3 days, depending on when I'm sending results. Any question about that? So you do need to look up the procedure for when you request a test. And so at some time, you should look up the procedure for the gelatin hydrolysis test, for the gram stain, and for the PR lactose test. Oh, there's something I forgot. Let me write that down. I forgot to mention to the other lab. Remember, for you, right? Yeah. That is the, I forget to tell them. You just started talking about what the heck I was trying to think. Now I'm gonna not remember what to tell you guys too. Um, hmm, Does it have something to do with the incubation time? Ah, I'm drawing a blank now what I need to talk about. Ah, back and see if I can figure out what I was talking about here. I was talking about the Simmons citrate test and telling you that it takes 2 days to grow. Oh well, we're gonna have to skip it because I don't remember what I was thinking. Um, and that means you won't get it either, which means I'll have to try and remember what the heck I'm supposed to tell you guys and tell both labs, which is probably better, and that way you'll get it at both at the same time, or at least the same day. All right, any questions about that part of the project? I still have a lot to tell you about the project. All right, let's go on talking about the project. You can read about the rationale, the project format. Um, you need to be following dichotomous logic, where you essentially run one test, get the results, look at your results in the table, and then pick another test. If you don't use dichotomous logic, this project will be penalized, meaning you'll get a markdown on the grade for this project. Everything's going to be done virtually, so we're just going to pretend that you're doing this in the lab. And you're going to be requesting different tests to try and identify your species. You should probably also, when you're talking about this project, both in your unknown notes and in the unknown report, meaning when you write up the report, you should discuss that I sent you the number tonight. We were going to pretend that I do that. You're not going to get it until next Tuesday so that I can reduce the number of contacts I have between me and my students. But where you pretend that you're streaking it out. So I gave you the unknown number, which represents your unknown species, and then you streak it out for colony isolation. Take cells from an individual colony, streak it out a second time, and then take cells that have gone through two colony isolation events, meaning on the second plate, you take cells from an isolated colony to set up your stock culture. And that way you're certain, not certain, but over 95% certain that you have a pure culture. And you need to do that if you were actually doing this in the lab, because when we sent you the cells in the lab, you would get two different species, and you have to streak it out to get a pure culture. Now, in the virtual lab environment online, we decided we're only going to send you one number and you will get a pure culture, but you should go through the motions as if you were setting up a pure culture because you should know how to do that and realize it's extremely important in microbiology. So you should streak it out 2 times and then use those cells to set up your stock culture for all future labs that you'll request, meaning you'll pretend you're going into your stock culture to use those cells to run the next test you set up. Any question about any of that? All right, so run your test. Use dichotomous logic. If you use logic that's not dichotomous, the project will be penalized. The penalization will be greatest if you use shotgun logic, which is simply run a whole bunch of tests at one time, hoping that something hits, sort of like shooting with a shotgun, and you send the bullets everywhere, and so you're hoping to get the duck. You should write down daily lab notes. These should be in your unknown notes. I'll show you that in a minute. You should write down your detailed notes for what activities you did on that day, what your observations were on the results I send to you. That is your results, your observations, and then your interpretation of those results. For example, if I send you a picture of a yellow tube, the result is that the test came back in a yellow tube. The conclusion or interpretation is the test was positive. Okay, so you should have both your observation, that's the result, and your conclusion, and that's the interpretation. You should also, in your unknown notes page, write down the species under consideration, and then any logic you have for what test you're running next. And then at the bottom of the unknown notes page, write down your test request.
Will we have access to this document?
The unknown notes page or the document I'm showing you.
The one you're showing us?
Yeah, you already have access to this. I already talked about it. You were supposed to pre-read this before I was talking about this. And you can find that right there, unknown instructions.
Oh, that is the 2 pages. Okay.
It's more than 2 pages. It's several pages. I don't know how long it is. All right. So this will be a project where 75 points. You get 75 points for the unknown report. Your unknown report must be typewritten. Of course, it has to be sent in, so it has to be typewritten. And use 12-point font. I have trouble reading anything smaller than 12-point font. Do not double space. You should have your daily descriptions for what test you ran. Well, first put the date. You can put the time if you wish, and then what test you ran, and then the information you hope to gain from the test, your observations you made from that test, the conclusions you made from that test, and then the species under consideration. So that should be in every day. You should not omit any data to make your project look better, and I can just look at your unknown notes pages if I need to. You may include drawings, photos, tables. They're very helpful. They're not required, but I will send you photographs for your test results. And you can include more photos that you can find online. Like, for example, I'll send you the test result, and let's say I send you something that's, I don't know, positive. You can find a negative test result online, and you may add it if you wish. Realize that whenever you're using a photograph that I sent you, you don't need to say supplied by Dr. John Volpe or Professor Volpe. You can just submit the photograph. I know where it came from because I sent it to you. But if you find any picture you want to add to your report online, you must put underneath the photograph where you got it, meaning what page you got it from. Otherwise, it's plagiarism. It's also a copyright issue if you don't do that. And then if you do put a photograph, you should put that in the citation, once again, where you got that page from, photograph from. Any question about any of that? Like I said, drawings, photos, and tables are not required, but they are very helpful. You do not, when you list the test, you do not need to detail the step-by-step details you did to run the test. For example, if you're running the gram stain, all you need to do is say, I ran the gram stain, and here are my results. The cells were purple, roundish cells, or purple cocci. And the interpretation is gram-positive cocci. The thoughts and words of your report should be your own. If ever you use words or advice from someone else, the instructor, other students, or get it online, you should put it in quotes and where you got it from. And that's whenever you're using more than 3 words of somebody else's words. And that does include AI. Remember, you're allowed to use AI, but if you use it, you should put it in quotes and say where you got it from. Otherwise, it's plagiarism. It is mandatory that in this report, meaning in the lab report, not in the unknown notes, that you put a table outlining the process you use to identify your unknown organism. An example table is in this document. So let's scroll down. So initially, the professor, in this case, the student, had all the species, and the first test was the gram test. And he got the result for the gram test, gram-positive coccus, and it narrowed it down to these species. The next test was that this person ran was the catalase test. The results and conclusion were catalase positive, so I guess that's a conclusion. The results for the gram stain would be you had purple spherical cells. For the results for the catalase test, you saw bubbling. So catalase positive, and that narrowed it down to 4 species. This is a table that was included in the student's report. And then the PR mannitol test was done next, and it narrowed it down to 3 species. The PR glucose test was done next. We have an unstable environment. Just a minute. Okay, did you guys not hear part of that? All right, PR glucose.
All of it.
I'm sorry?
I heard all of it. Sorry.
Okay. All right. I saw a message, so I stopped talking when it was unstable. PR glucose narrowed it down to 2, and then the student did the citrate test, and it narrowed it down to 1. So you should have either a table like this, or you can make a hand sketch diagram. Okay? Most students use a table, but you could do a hand sketch diagram and then include that diagram in your lab report. But you must detail how you obtained your unknown species down to the species you think it is. That is required. It's also required that once you have it narrowed down to 1 species, you give a microorganism paragraph describing the microorganism, including its diseases, its mode of transmission, common sources, places where you find it, different treatments, and any other interesting information. This paragraph only needs to be 1 paragraph long, but it can be 2 paragraphs. Do not exceed more than 1 type written page discussing your microorganism. If it's more than 1, I'll dock you. It must be 1 paragraph. You must have a citation for at least where you got the information from for your microorganism paragraph. So there must be in this project at least 1 citation. And if you have pictures you got from the internet, you'll have more than 1 citation. Of course, you could have more than 1 citation for your microorganism paragraph as well. So you should have a literature citation page at the very end of this report. Any question about any of that? All right. So I do take plagiarism seriously. No plagiarism is to be allowed. Your report will go through a plagiarism checker. And if plagiarism detected, I'll subtract at least 5% minimum. And if it's a gray, just plagiarism, I could subtract all 75 points. The grading criteria follows. I have a grading sheet in the lab. You're going to be mostly graded on the logic you use for performing which test. There will be some points on if you get the correct species at the end. It's no more than 10. You'll be graded on the completeness of the report. The organization, clarity, and neatness of the report. Graded on, let me see where I've got it here. Oh, I'm in the wrong place here. On the grading rubric. One I want. What did I shut down? Hopefully I didn't shut you guys down.
We're still here.
Yep, I see that. All right, I shut that down. Let me try again. Might have to go online if I'm not finding it. That's the key, notes key. Oh, that's it there. Okay. I should call that rubric because that's what I was thinking. All right, you can find the grading rubric. There it is, unknown report grading rubric right there, which I'm showing you here because it'll display better than in Canvas. You'll be graded on the utilization of resources. Was any excess media used or were any irrelevant tests performed? If there's none, you'll get 10 out of 10. If there's one test irrelevant or excess media, you'll get 8 out of 10. If there's two, you'll get 6 out of 10, etc. Was dichotomous logic used in selecting your tests? No excess tests, you get 10 out of 10. If you have one excess test, you get 7 out of 10. Two excess tests, 5 out of 10. If you use shotgun logic, shotgun approach, you'll get 3 out of 10. No logic used in selecting your test, 0 out of 10. Were all relevant tests used? Meaning, were you able to successfully identify your unknown species? Or did you need to run more tests to identify your unknown? No tests were omitted, 10 out of 10. If you needed to run one more test to be certain of your unknown, then it would be 8 out of 10. Two tests a minute, 6 out of 10, etc. Were the correct species included at each step? So in your unknown notes, as well as in your unknown report, you should be listing the species in consideration at that point in your unknown test. Like at the moment, you don't need to write this down because I know that for a fact, but at the moment, all the species under consideration are all the species shown in this table. For the gram positive and the gram negative, it's something like 20 to 24 species. Okay? So you don't need to do that because these are all the species. But after I give you the gram stain, you should state, saying it's gram negative, you should state that all the gram negative species are under consideration. And because I'm going to give you the gram stain, the gelatin hydrolysis test, and the PR lactose test all together, in your unknown notes page as well as in your report, you could say, these are the species under consideration after the gram stain. These are the species under consideration after the gram stain and the gelatin hydrolysis test. And these are the species under consideration after the gram stain, the gelatin hydrolysis test, and the PR lactose test. You could do it 3 different times, or you could just say, these are the species under consideration after these 3 tests, meaning you only do it 1 time at the very end. That's fine. Every other time you will be requesting 1 test, in which case you should give the species under consideration after that 1 test. And if you get all the species correct, you'll get, where do I have that? You'll get 10 out of 10. If 1 species is incorrect out of all the species you mentioned, it's going to be 8 out of 10. 2 to 3 species incorrect, 6 out of 10. Over 5 species incorrect, or if you do not list the species under consideration, you'll get 2 out of 10. Completeness of report. All information is presented, 10 out of 10. 1 instance of missing information, 8 out of 10. 2 to 3 instances of missing information, 6 out of 10. Effective and clear communication, meaning your English, grammar, spelling, and punctuation all good. If everything's excellent, you'll get 15 out of 15. A few errors, but logic is understood, 13 out of 15. Some errors, but logic is still understood, 11 out of 15. Some errors, but I can't understand some of them, 7 out of 15. Identification of your unknown. You got it correct, you'll get 10 out of 10. If you get the species incorrect, but you're within the same group or type of cell, you get 7 out of 10. If you are outside the group, but the gram stain is correct, you get 5 out of 10. If you identify a species and it's the incorrect gram stain, you'll get 0 out of 10, for a total of 75 points. Any question on the grading of this project? All right, let's move on. Actually, this is telling you how you did it in the lab. We gave you a tube. You then streak it on 3 different plates in the lab. See the growth on these 3 plates. And you usually streak it 1 more time on TSA or a nutrient auger to get cells that went through 2 colony isolation events. And then you set up your stock culture. And then you use the stock culture to obtain your cells for all future tests. We're going to pretend that you're going to do that. Any question about any of that? So in your lab report, you should be discussing that you got the bacteria, you streaked it out to get isolated colonies, you harvested cells from 1 isolated colony, streaked it out a second time for isolated colonies, obtained cells that went through 2 colony isolation events before you set up your stock culture. And you set up your stock culture from cells that went through 2 colony isolation events. And that way you're guaranteed of getting a pure culture at least 95% of the time, probably higher. The last 2 pages, or maybe the last page of this document, goes through the taxonomist process. If you want work or a better understanding of the taxonomist project, you can go through this example, go through the questions. Don't turn the questions in because I'm not going to grade it. I'm not going to even look at it if you turn it in. So you can work through the questions, and that will just help you about dichotomous logic. If you don't understand this project using dichotomous logic, please discuss it with your lab instructor, which is me, as soon as possible, because this project should be using dichotomous logic or you will be marked down. It's called dichotomous logic because essentially you're asking a question, all cells beginning with, sorry, you start with all cells or all species, I guess. And then you do the gram stain. And it'll separate into gram positive. Oop. And gram negative. And let's say it's gram positive, so we can scratch this one out. And then you do the catalase test, which will separate into catalase positive and catalase negative. Let's say it's catalase positive. So we scratch these out. And then you do another test, separating into essentially two different possibilities. A positive, up, that wasn't very good. Let's erase that. My mouse cord got stuck, and so I couldn't move my mouse clearly. And one result will be positive. Let's just write it. Up. One result will be positive. The other result will be negative. Dichotomous. Any question about that? All right. Let's talk about a few other parts of this project. After the first email that I'll send you, hopefully next Tuesday, all the information you do and all the communication between you and I will be handled in your unknown notes pages. So when I send you the, well, actually, you could start the unknown notes page today and say you are requesting the gelatin hydrolysis test on your unknown number. And I guess you can say you're requesting the number because you're not going to get it until next Tuesday. But you can open up your unknown notes page. And there's not much to see here, but let's open it up. I have a grading. You will have an upload page there. Let me shut this down and I will come to an unknown notes page in a previous term. It'll show you the grade, which is why I'm shutting it down. Let's go last term. Unknown notes page. Go to the student. I got to remove the grade so you don't see it. And now let me share my screen. So this student actually has more than they need. They got it looks like they're starting to write their report in their unknown notes page here. But unknown number 61, I should probably blank out the student's name too, but Too late. And then on November 12th, she performed the gram stain. And there's the results of the gram stain. She should put, oh, there it is, red, pink color, meaning that that's the results. And then the conclusion is gram negative. She should put that conclusion gram negative because she's got it under the observation, but you should have the observations, the activities for the day, and that's sort of what that is. And she should have the conclusion for it's gram negative. Any question about any of that? And then at the end of the report, end of the day here, I got that down already. So coming here, this is the gelatin hydrolysis test and the PR lactose test. And she's got the possible species under consideration here. And then at the very bottom of the unknown notes page, you should have the next test that they're requesting. And this student is requesting the methyl red test as the next test. Um, the um, the test request must be at the bottom of the page, or you can have it at the very top of the page. I'll see it here, and or I'll look for it in the bottom of the page. If you have your test request someplace in the middle of the page, I will not get your test request until I actually read the document. And when I'm looking for unknown tests, meaning an unknown test request, I'm going to simply look at the bottom of the document. So put it at the bottom of the document, your next test request. If it's at the top of the document, I will see it. But if it's in the middle of the document, I'm not going to see it until I actually read it. And I might not read it for several days. So put it at the bottom of the document, or if you really want, you can put it at the top of the document. Anywhere else, no guarantee I'll see it for several days. Any question about that? All right, let's go back to the unknown notes page. This page is for your unknown project. So please ask if you get confused about it. The project will be done online. After the first one or two emails from me, record all your results and interpretation in these unknown notes page here. Then record your daily activities, especially the test request you make for your unknown project. Put your test request at the bottom of the page. Everything you do concerning the unknown project will report it here all in one place. And that'll make it very easy for both you and I to look up the data. You should have, I don't see that in there, but you should have your species under consideration after you put your test results and your test conclusion. Do not use the comment section. Oops, right here. One of these places. Do not put your data. Oops, shouldn't show you that. Do not put anything you put here. If you want to put a comment, put it in your document. I will read it. If you put it over here, I'm going to delete it because I want this region for me. And as you can see, I got a lot of comments in here, and I'll put all the results in the comment section. Okay, so even this student here has a number of comments. If you put your comments in here, it'll just be too much. So don't put any of your comments here. Put it in the document. I will read it. Any questions about your unknown notes page and what you do to get it to work? Like I said, initially I picked the 3 tests for you. I will send you those results in an email. That will be the last email between you and I on this project. If you send me an email requesting a test, I'm going to tell you that I'm not going to honor a test request in an email. If you want a test request, put it in your unknown notes page. Any question about that? All right. So that's your unknown notes page. Let me stop the share. I got to put the grade back in the student and save it. And then I'm going to get out of here. Pull that down. All right, let me share my screen. Oops, wrong one. All right, so I've talked about the unknown notes page. By the way, the unknown note page will be graded. It'll be out of 5 points. I won't get that graded until after you've turned in the project. Probably not until finals week is when I'll probably grade the unknown notes page. It'll be worth 5 points, much less than the unknown project, which is turned in. Let me see. The unknown notes page is turned in. Excuse me, the unknown notes page, the last day you can work on it's March 7th. The unknown report must be turned in March 8th. The unknown report, 75 points. The unknown notes, 5 points. The unknown notes will count as one lab grade, 5 points. Talked about the rubric, unknown list, the building a dichotomous game plan. I've already told you about dichotomous game plan. But if you need more help on dichotomous game plan, you can click that link and read it. Let's talk about the sample journal. Before you do your lab report, you should look up the sample journal. And I'm not going to show it there. Let's go here. It'll display better if it's not in Canvas. This is a sample journal that's actually was written by an instructor. And I remember what I did last, and I got to change that. I guess I'm not going to change it. Anyways, in this journal, the instructor has gone through and look here, moving it down. Let me delete this and start all over. Because the things are on top of each other. There we go. So he's got the unknown number here and his name, the class, the term. And then the first day, he streaked out the unknown species on two 2SA plates and two EMB plates. Got the growth here. Picked one of the colonies, colony B there. She doesn't have very well isolated, but streaked it out again. Made observations. And this looks like about the first day of real results. So in his unknown notes page and in the unknown report journal, or the unknown report, he wrote out day 4, February 6th, observations he made on this day. Colony B reserve slants had homogeneous opaque yellow growth, meaning that it's uh, yellow pigmented bacteria. So that's an observation he made that's useful because that tells you the pigmentation, and there's only a few species which are pigmented. He then did the activities on this day. He did the gram stain on colony B, saw the results. The cells stained purple, they're spheres. The conclusion, interpretation of the results. The appears my cells are gram-positive cocci, and there's this picture there. On day 5, he has no observations because you normally do not get your results on the same day you run the test, but with the gram stain, you often do. You won't because I'm going to have you make the gram stain on Sunday and I won't give you the results until Tuesday. So I'm pretending you're looking at the slide on Tuesday. On day 5, observations none. And then here's the test he ran. He ran the catalase test. This actually gets the results on one day. So we saw bubbles for the results of the catalase test, which should be under the observations actually on that day. And then the conclusions, the colony B is catalase positive. And then here he's got it narrowed down to what families were under consideration, obviously the gram-positive cocci, and then narrowed it down to family Staphylococcus or Micrococcus. You could list out the entire species. He's not done that. He might do it in the table. All right, any questions? You should look at the sample journal before you write your lab report. The lab report in the sample journal is one done by someone doing the work in the lab. Obviously, you're going to be doing all of your work online. You request a test, and then I'll show you pictures of the results. You have to make the conclusions. And there he's narrowed down the species, Staphylococcus aureus. By the way, let me state that he actually narrowed it down right here to Staphylococcus aureus. And you are allowed to do one test extra. Let me mention that to my other class. You are allowed to make one complementary test, meaning where you confirmation test. Sorry, one confirmation test. You are allowed to do one confirmation test to confirm that it's Staphylococcus aureus, which actually he got right there because these species weren't under consideration before. So you're only allowed to do one extra, and he actually did one, two, three two extra. So you're really only allowed to do one extra. If you do more, you will be penalized, but you are allowed to do one extra test to confirm that it's the species that you think it is. Any question about any of that? All right. And there's his paragraph about his organism that he found. And then there's the references. Any questions about this project? All right. If you have any questions, email me. I will strive to help you. I've still got one thing to tell you. It's just that I don't remember what it is. And obviously I forgot the other class. And hopefully I'll figure it out. But there's three things I realize I need to tell the other students, and that is write their unknown requests at the bottom of the unknown notes page. I won't honor a test if they request an email. That one I'll just send to them if they ask a test in an email. And I need to tell them that you are allowed to do one confirmation test after you've identified your unknown species. And that's just to confirm that you have the species you think you have. You do not have to do the confirmation test if you're confident you have the species you think you do. You can end the project there. All right, any questions? All right, that's it. I will see you next Thursday. Bye.